I'm going to talk about is whether a deficit in hippocampal interneuron function can actually be considered as a primary deficit or a primary pathology in schizophrenia. And this is all data we've done in preclinical models. So just as a bit of background, schizophrenia is obviously a debilitating disease. It affects up to about 1% of the population, and it's characterized by three specific symptom classes. The first being positive symptoms, which are the more well-known symptoms. These include things like hallucinations, delusions, and psychosis. And the other two are less well-known, but I guess equally as debilitating. These are negative symptoms, including things like social withdrawal and anhedonia, and also cognitive dysfunction. We see deficits in working memory. So we can model schizophrenia somewhat in rodents. There's a number of different preclinical models. The one that I use is known as the MAN model. It was generated in the lab of my postdoc advisor, which is, I guess, why we use it. And it involves injecting pregnant female rats, specifically on gestational day 17, with a mitotoxin known as methylase oxymethanol acetate. And we use saline injections for control purposes. And what this produces is alterations in the offspring that are somewhat analogous to those observed in human schizophrenia patients. And for the sake of brevity, I'm not going to go into specifics. It's been extensively published, but we do see histological changes in this model that mirror that seen post-mortem in patients. We see neurophysiological changes that mirror that seen with imaging studies in patients. And we obviously see behavioural changes that are somewhat analogous to those seen in patients as well. So we think it's a pretty good model as far as models of psychiatric disease go. So the primary technique that we use in the lab is in vivo extracellular recordings, primarily from the dopamine neurons found in the ventral tegmental area. So we do these all in chloral hydrate anesthetized rats, and what we do is we grab a glass microelectrode and we put it in the ventral tegmental area, and we identify dopamine neurons specifically based on their electrophysiological characteristics. The most prominent one being this long duration action potential. We also have a very irregular firing pattern. So once we find these dopamine cells, we're interested in three distinct patterns of activity. The first is the number of cells found per electrode track. And so the current hypothesis is that about 50% of the dopamine cells in the ventral tegmental area are silent. They're not spontaneously active. And this is due to an input from the ventral pallidum causing them to be hyperpolarized. We think the reason for this is that it provides a gain of function. So you can now either increase or decrease the number of cells that are spontaneously active to change the gain of the dopamine system. So this acts sort of like the volume knob on an amplifier that you can turn up or turn down the dopamine signal based on things such as novelty or an environmental context. We also look at the firing rate and burst firing, the sort of more typical characteristics of neuronal activity. But for the sake of today's talk, I'm not going to focus on those, as I'm pretty sure none of our manipulations actually change firing rate or burst firing. So what I'm going to be talking about is this sort of parameter of dopamine neuron population activity. So just to briefly summarize the work I did as a postdoc in Tony Gross's lab at the University of Pittsburgh, we have the sort of the schematic of the dopamine neurons in the VTA of a normal animal, where we have about 50% of them that are spontaneously active, and we have a variable gain on the system. We can turn this up or down based on context and other things. In our model of schizophrenia, what we found is pretty much all of the dopamine cells are spontaneously active. So what we think is happening in our model of schizophrenia, and potentially schizophrenia in humans, is that all the stimuli that come through get ascribed this really, really high dopamine signal. So they all come through with this enhanced salience. And these are including stimuli that are really not salient stimuli. And we think the consequence of this is that you end up with paranoia, delusions, and hallucinations. Some of the other work that I did demonstrated that this was directly attributable to increased activity within the ventral hippocampus. And so our current hypothesis is that the pathology is up there in the hippocampus. You have enhanced activity that drives this dopamine hyperfunction. It does so via a multisynaptic pathway, including the nucleus accumbens and the ventral pallidum, essentially disinhibiting those dopamine cells. And so this is the data, and I'm going to be showing you a lot of graphs that look like this. On the y-axis, we have dopamine neuron population activity as number of cells per electrode track, and these are our control animals, and this is our model of schizophrenia. And what I just mentioned before is that we have this significant increase, a twofold increase in the number of spontaneously active cells in our model of schizophrenia. And we know this is due to aberrant activity in the hippocampus, because if we inactivate this, and in this case we use tetrodotoxin as a non-specific inhibitor, we can completely reverse this elevated dopamine neuron population activity. What's cool about this is it's consistent with recent imaging data in human schizophrenia patients demonstrating hippocampal hyperactivity at rest, which is correlated with positive symptom severity using PPRs. So we sort of had this understanding there's a dopamine hypothesis in schizophrenia, too much dopamine is bad, there's no sort of pathology there. We've now identified the hippocampus as a potential site of pathology, but just saying it's overactive really doesn't get to the reason why. So we're really interested in trying to get to the bottom of it. And while I was at Pittsburgh, David Lewis, who's also there, published some data looking at post-mortem human schizophrenia patients' brains. And what he saw was a significant reduction in the expression of a calcium-binding protein known as parvalbumin. And parvalbumin is found in a subclass of interneurons that are found throughout the cortex and the hippocampus. These parvalbumin-positive interneurons are perisomatic targeting, they're fast spiking, and they're really well situated to regulate pyramidal cell activity. So the sort of hypothesis is that you get a loss of function of these cells that results in enhanced hippocampal output that then results in enhanced dopamine system function, which is underlying the behavioral deficits that we see. So we had a look in our NAM model to see if we do get decreases in parvalbumin expression. We first did immunohistochemistry, and we saw decreases in parvalbumin-positive cells throughout the medial prefrontal cortex and also the ventral hippocampus. This is region-specific. It wasn't found in some of the other regions we looked at. More recently, we've replicated this using Western blot. This is work by Li Chan, who's a postdoc in the lab, and she's looking at the man phenotype across development, starting I guess just after birth all the way through to adult, and she sees the decrease in parvalbumin expression appearing around about postnatal day 25 and continuing through to adulthood. So that's all well and good, but it's really correlative. We see a decrease in parvalbumin expression and an increase in hippocampal activity. We want to know if the loss of interneuron function is actually sufficient in and of itself to change hippocampal activity and then downstream alterations in behavior. So to do that, we took advantage of a pretty unique characteristic of these parvalbumin-positive interneurons, and that's that they're surrounded by a highly specialized form of extracellular matrix known as a perineuronal net. And this is work done by Mishi Shah when she was at Tech in the lab. This is immunohistochemistry looking at parvalbumin-positive cells in the ventral hippocampus, and this is staining for the perineuronal nets, and you can see there's a high degree of co-localization. And these nets are thought to support the high firing frequencies of these interneurons. So our idea is that if we can degrade these perineuronal nets, we'll lose the phenotype of these cells, and then we can look downstream and see what the consequence of that is. The way we degrade these nets is by administration of an enzyme known as chondroitinase. Chondroitinase comes in, chews up these chondroitin sulfate glycosaminoglycans. Basically, these molecules support the structural integrity of the perineuronal net. If you digest these molecules, you lose net function. So what Mishi did is she injected chondroitinase in the ventral hippocampus, and what we saw is a complete loss of perineuronal nets throughout this region. As a control, she injected another enzyme. She used penicillinase into the ventral hippocampus, and you can see that you still have perineuronal nets under controlled conditions. So the first thing we did was recorded the activity of pyramidal cells in the hippocampus to see if this actually did affect hippocampal output, and indeed we see an increase in pyramidal cell firing in these chondroitinase-driven rats. We then look downstream to the dopamine system, and what we see is a twofold increase in dopamine neuron activity, exactly the same phenotype from a neurophysiological standpoint that we saw in our man model of schizophrenia. So it's all well and good to show electrophysiological data and say we see changes in activity, but I guess what's ultimately important is whether or not that changes behavior. And so one of the behavioral approaches we use is quite a simple test. It's amphetamine-induced locomotor activity, and it's based on the observation that human schizophrenia patients display an enhanced response to psychomotor stimulants, including amphetamine. This is distance traveled on the y-axis, time on the x-axis. You see a locomotor response to the novel environment. Once that subsides, we inject amphetamine at a low dose of 0.5 mg per kilogram, and you can see this locomotor response in response to amphetamine. This is data from a control animal treated with penicillinase. When we look at our rats that were treated with chondroitinase that had this loss of interneuron function, the hippocampal hyperactivity and the enhanced dopamine neuron activity, we see an enhanced response to amphetamine administration. It mirrors that which we see in our model of schizophrenia as well. This is just an aside. Some of the work that's currently being done by Angie Boley is trying to address this more directly. She's done some studies looking at lentivirus administered short hairpin RNA targeting parvalbumin to see if a loss of parvalbumin itself is sufficient to produce these same deficits. And what she's shown so far, this is really preliminary, is an enhanced response both at a low dose and a higher dose of amphetamine. We now include the Dr. Fran's dose of amphetamine, so we have a dose-response curve to present. So all of this sort of comes to the suggestion that a loss of interneuron function is sufficient in and of itself to produce the phenotype that we see in the man rat. And that led us to the idea that restoring interneuron function in our model of schizophrenia may actually reverse some of the deficits that we see in this model. So to do this, we used a cell transplantation technique. We dissected out the medial ganglionic eminence of embryonic rats on embryonic day 14 and a half. The medial ganglionic eminence is the primary source of developing GABAergic cells in the developing brain. And then later on in development, these migrate and form interneurons in the cortex and the hippocampus. So we dissect out this medial ganglionic eminence, we dissociate these cells, and we inject them into the brains, into the hippocampus of control or MAM-treated rats. We then wait 2 months for those cells to sort of migrate, survive, and sort of form part of the anatomy of the brain. The rats that we use as donors to get this tissue from are green fluorescent rats. So these express the GFP on the ubiquitin promoter, so all the cells that we obtain are GFP positive. And we do this so we can follow the fate of these cells and see what happened to them after transplant. And we're interested in whether this can rescue signaling in this man model of schizophrenia. So this is one work done by Stephanie Perez, and these are some histological slides taken 2 months after transplantation. And what you can see here by the arrows are MGD transplanted cells that migrated quite extensively through the hippocampus. This sort of shaded area shows the distribution of the migration. They tend to migrate quite extensively. You can see they've obviously survived. This is 2 months after transplant. If you look at higher magnifications of these, they have dendrites and axons. They look like, at least anatomically, they've integrated within existing circuitry. She did some double-labeled immunohistochemistry to confirm the phenotype of these cells, and a majority of them were double-labeled for glutamic acid decarboxylase, showing they were indeed GABAergic cells. She also stained for parvalbumin, and a subpopulation of these are parvalbumin positive as well. So what's the consequence of this? So this is data in the ventral hippocampus looking at pyramidal cell activity. The difference here is showing the effect of MAM. So this is something that we've published previously, showing enhanced hippocampal activity in our model of schizophrenia. If we look at rats that have had the cell transplants in the ventral hippocampus, you can see we restored this aberrant activity right down to the same level we see in control animals. If we look at the dopamine neuron activity, which is downstream of the hippocampus, again, MAM-treated rats had this twofold increase in dopamine neuron population activity. And in the rats that had the cell transplants, we can completely reverse that as well. So again, it's good to show the electrophys data for what happens with behavior. So here's the locomotor response to a low dose and a slightly higher dose of amphetamine. You can see that, as we published previously, the MAM-treated rats have an enhanced locomotor response to amphetamine. And in rats that had the live cell transplantation, this is completely blocked. And what we have is essentially the MAM rats with the cell transplants are superimposed over our saline-treated animals. So taken together, we've shown that our MAM model of schizophrenia displays this enhanced hippocampal drive of the dopamine system. We've shown that a loss of interneuron function in and of itself is sufficient not only to augment hippocampal activity, but also to increase dopamine neuron activity and to alter behavior. We then use this information to show that transplantation of inhibitory neurons back into the hippocampus can actually restore both the neurophysiological and also the behavioral deficits in our model of schizophrenia. And it's led to our suggestion that targeting hippocampal transmission may actually be a novel therapeutic approach to treat schizophrenia and also potentially other psychiatric disorders associated with the dopamine hyperfunction. And we've actually shown this with a couple of different ways. We've looked at allosteric modulators of the alpha-5 subunit of the GABA-A receptor and shown them to be effective in this model. Stephanie has published a paper recently looking at deep brain stimulation of the ventral hippocampus, and that seems to restore functioning in these rats as well. And we're currently doing some studies with Andrea Giuffrida and Alex Zellier, and we have initial data looking at the increasing endocannabinoids being potentially beneficial as well. So with that, I'd like to thank the lab members, particularly Stephanie, Angie, Mishi, and Lee, who did the majority of the work, financial support. Thanks for your attention.
By the way, do you see changes in the neuroregulin signaling in these models?
We haven't looked in this model. Obviously, neuroregulin, there's a big literature looking at neuroregulin schizophrenia. We haven't looked at it in this model. Yeah.
From your recent report, it seemed like the increased dopamine is also responsible for the negative symptom like social score.
It may be. Yeah, it may be. So one of the things that we've focused on primarily here is indexes of positive symptoms. We're starting to branch out and look at cognitive symptoms, and Stephanie's shown that with deep brain stimulation of the hippocampus, we can actually restore the cognitive deficits. We're starting to look at negative symptoms as well, and at least in our hands, we haven't got any conclusive data. Whether that's due to sort of having this constant dopamine signal that you can't modify anymore, you now can't have a sort of elevated dopamine signal in response to a rewarding stimuli because everything else is at that level. So it may be that sort of constant affect may underlie negative symptoms as well, but we have no data to demonstrate that.
You were talking about the net, which is fascinating. Do you know whether the net are effective in schizophrenia or in the model? And knowing that they are not present in juvenile animals, and you see when you show the probability alteration in your model, there was nothing in juvenile, and it didn't increase as much in your lab compared to the wild type. Do you know whether the net phenotype is the same?
Yeah, we don't know about net phenotype across development. We do know about the net. So we did Western blot for markers of proteins that are components of the net, and we see in our model of schizophrenia a decrease in the expression of some of those markers. People have also looked at post-mortem human tissue, and it was sort of a serendipitous discovery. They were looking at glial infiltration into, it was temporal lobe, it wasn't hippocampus specifically. And as an aside for that, they'd stained for these perineuronal nets. They sort of mentioned in the discussion, by the way, we saw a loss of perineuronal nets in post-mortem human schizophrenia patients. So yes and yes to the model and post-mortem patients.
What does MAM really do in the circuitry of critical rhythm?
Yeah. So if you'd asked me that 3 years ago, I would have said it's a mitotoxin, it inhibits cell division, and that's what's, we give it during development, that's what's causing the phenotype. We did some studies with BRDU that we haven't published, and there's also been cell counts in the animal. We really don't see cell loss in this model. So we're starting to rethink what it's actually doing. So we don't think it's acting as a mitotoxin per se. We've done some microarrays looking at methylation. It's a potent DNA methylator, and we see differential methylation of promoters of genes that are associated with schizophrenia in humans. Whether or not that's responsible for the phenotype, we don't know. Stephanie's got a poster showing that this phenotype is heritable, so we're doing something that is somehow inherited through the germline. Methylation is one idea, but there's many others that we could guess at.
You don't think the connections are changing at some key development time point between the
Yeah, so that's initially why. So if you give this drug at embryonic day 15 or at a different time point, you get microcephaly, you get a whole host of characteristics that are not analogous to that seen in schizophrenia. It was originally chosen at gestational day 17 because most of the subcortical regions are fully developed, and you've got some in the temporal cortex regions which are associated with schizophrenia. It also goes with the developmental disruption sort of theories of schizophrenia with prenatal infection and things like that. So that was initially the theory, that we're affecting the temporal lobes and the parts of the brain that we know were affected in human patients. It turns out that what actually may be happening may be a little bit, it's not that simple. Yeah.
Are you able to differentiate like some type of neurons that are
I guess we're able to, but we haven't. So, yeah, I guess in patients, David Lewis has obviously done a lot of work looking at basket cells, and not just Hippocampal cells, but CCK and NPY and the different types. The reason we're looking at subpopulations of interneurons is he showed that you don't get sort of a loss of interneurons in and of itself. So it's a significant loss of a subtype of interneuron. If you look at all interneurons, you don't really see changes. But we haven't subtyped them at all.
So, with your model, like you said, it had to be given during a specific time where orthorhomatosis corporate protects, because obviously it's going to problem cells that are supposed to be dividing, not dividing.
Yeah.
But did you, and you didn't see, beginning with the DRD studies, you didn't really see that there was an effect of cell death. But did you look at the specific areas where we know that neurogenesis is occurring at those places, so like for in the hippocampus or in the subventral zone where we expect that there are cells undergoing a lot of disease in those areas, even at that point?
Yeah, so they've looked at prefrontal cortex and hippocampus in terms of actual stereology and cell counts. And we really don't see big changes. What we do see is heterotopia. So we see a disorganization of hippocampal structure. And that is consistent with what you see in patients post-mortem as well.
All right, great. Before we break for lunch, let's thank Dan. We have two announcements. Lunch will be out in the area where we have breakfast. And we want to make sure we have 1 hour, and we're going to start the next afternoon poster session on time. So during that hour, if the afternoon posters presenters can get their posters up and the other ones have to take them down, okay, put them up if they're not.
Before you come back in here.
The second announcement was totally incomprehensible to me, so I'm going to let Bethany make it.
So all the lunches are set out as boxes, just for efficiency's sake. All the boxes should be the same in there except the actual sandwich. So there's a unforgettable is on the far left, like towards the patio, and that means turkey. That doesn't mean turkey to me, but it does to druthers. So there's turkey, there's ham, there's like a roast beef, there's closest to the drinks is the vegetarian boxes that are salad. So if you're inclined that way, then there you go.
Thank you.